All right, so this week we are beginning our final system, essentially, of this part of anatomy and physiology. So our entire third unit of the course is going to be focused on the nervous system. So this week we're just simply introducing you to nervous tissue, the structural organization of kind of the basics of the nervous system, how we divide the nervous system into the central, peripheral, autonomic, what those terms mean, the cells of nervous tissue, and then we'll begin to talk about membrane potential, graded potentials, and action potentials, essentially how signals and communication are facilitated through nervous tissue. And so that's beginning here in chapter 12, and then we'll later expand and build on this throughout the rest of this quarter as we learn more specifics about the spinal cord and about the brain and about the various nerves that come from each of those parts of our body, how they integrate sensory signals, and how we send back out motor signals to control our bodies, et cetera, et cetera. But for now, just starting to get an introduction to these things and these ideas. So referring to the nervous system is a bit rudimentary. We have a lot of different levels of organization or categories that we provide through the nervous system. So we have our structural organization of the central versus peripheral nervous system. We have various nerves that make up that peripheral nervous system, which we'll talk about more in detail in later chapters. And then we have functional separation. We have our sensory and motor divisions, aka our afferent and efferent divisions of our central and peripheral nervous systems. And then we have somatic versus autonomic, meaning what is consciously being sensed and controlled. So perception of our sensation would be conscious perception, that is somatic. And then autonomic, which things are being automatically perceived or not perceived, sorry, automatically sensed and controlled without your conscious perception, et cetera, et cetera. So a few different types of division to discuss. We have various cells, looking at this as its own type of tissue, nervous tissue. So we have cells like neurons, which are mainly the ones sending signals, and then supporting cells, which are our glia or glial cells, ganglia. And those look and act a bit different depending on if we're talking about the central or the peripheral nervous system. And then, like I said, our very last topic is going to be to discuss potential. So our resting membrane potential, graded potentials, and action potentials, how our nervous tissue sends communication, sends signals to other neurons and to things like muscles and the rest of your body. So the structural classification of the nervous system splits from the central, which is literally in the center of our body, composed of our brain and spinal cord. That makes up our central nervous system. These are our processing centers for information. So this is going to be the source and final destination of, sorry, let me back up. This will be the final destination for sensory information, and then the source of all motor information is coming from our central nervous system. Again, the brain and spinal cord. And then all the neurons that leave the central nervous system, either directly from the brain or further down the body from the spinal cord, that makes up what we call our peripheral nervous system. So once they have left the central nervous system, they will be bundled into structures called nerves. And so we'll talk about all of our different specifics about the types of nerves later, but that is generally that organization. So the central nervous system is like the central hub, the headquarters for all of your neurons and parts of your nervous tissue. And then once these neurons leave the central nervous system, that has now made up our peripheral nervous system, which spreads out throughout the rest of the body. And they will do that in these organized structures called nerves. So we can further categorize these peripheral nerves as to where they're leaving from the central nervous system. So we'll have cranial nerves, which are coming directly from the cranium or the brain. And then we have our spinal nerves, which are all those that are leaving or entering the spinal cord itself. So we have many more spinal nerves than we do cranial nerves, and we will talk about all of these again later in the course. So that was the structural division of our nervous system, and then we have a functional division. So the major functional divisions are between sensory and motor. So again, afferents, so the effect sensory is then going to cause an effect, which is going to be a motor response. So cause and effect. Sensory is the cause that is then going to turn into some effect, which is going to be motor, some control in response to that. So we can talk about our afferent system and our efferent system, how we are receiving information and how we are sending that information, sending other types of information back to our body. And we also have autonomic versus somatic functioning. So a majority of what is happening inside your body, a majority of the information in general that your nervous system is dealing with is going to be autonomic, meaning it is unconscious. You are not consciously perceiving all the things that are happening inside of your body. And you're really ignoring even a lot of the somatic information that is going to your brain because we end up being habitualized in order to concentrate our focus. So you're really not consciously perceiving a majority of the information that your body is receiving, that it is sensing, and you're not consciously aware of a majority of the information that your nervous system is then sending back out to the rest of your body to control it. And so we'll talk about what both of those systems look like, right? How your body is naturally controlling itself and taking care of itself, and how our brain, this conscious awareness and perception, plays a role in the function of our body as well. Yeah. So, majority of the work that we're going to be discussing within the nervous system is being done by neurons, or this is our main area of focus. These are what are sending signals to and from all the various parts of our body and making up a majority of the nervous system, the actual functional part of the nervous system. And so a typical neuron is going to look like this. This is considered a multipolar neuron, and we'll talk about what that means in a second. But we can use this to discuss our major structural components of these cells. So this is one cell. These are really, really long cells. One neuron could run the length of the tip of your big toe all the way up to your spinal cord, just above your sacrum. Okay, so that is a very long distance for one single cell to travel. And most of our major neural pathways are only composed of 3 or 2 neurons. So going all the way from the brain to the big toe, or from the big toe to the brain, that is only going to consist of 2 to 3 neurons in either direction. And the same could be said for any part of your body. Most of these neural pathways are just composed of 2 to 3 of these cells. So they end up being really, really long. Structurally, a majority of the cellular components of a neuron are found in what we call the cell body, or the soma of that neuron. So this is our cell body over here. In our example multipolar neuron, the cell body has a bunch of these little branches coming off of it called dendrites. Okay, so they end up looking like trees. These are the receptive structures of the neuron. So this is where information is going to be received, and then information will be sent from this neuron down its axon, which is this much longer branch, which is going to end on some telodendria. So these are referring to the branches at the end of an axon, telodendria, where there are a bunch of neurotransmitters, those signaling chemicals that are sitting in vesicles. So packaging a bunch of these chemicals in these little knobs that we call synaptic terminals, or synaptic knobs. So the pathway of information in any neuron is going to come from the dendrites, and then through, or a lot of the time, I suppose, is going to come from the dendrites and then through the axon. Some signals are going to be sent directly onto this axon hillock. Some signals can be sent directly onto a synaptic terminal. There's lots of different levels to how our nervous system communicates with each other. But to put it most simply, you can think of this unidirectional method of information being sent from one axon of the previous neuron onto the dendrites of the next one, and then down the axon of that new neuron onto the dendrites of the next neuron, or onto whatever cell it's acting on, such as our muscle cells, our skeletal muscle cells that we were discussing in the last chapter. Okay. So that's the pathway of information. We have these receptive branches called dendrites, and then what is sending those signals down our axons to the telodendria, releasing neurotransmitters from these vesicles in our synaptic terminals. But going back a bit to the body, the soma of our neuron. So this is where you find a majority of the structures that you associate with a cell. We have our nucleus with all the DNA of the neuron. We have our Golgi apparati, our endoplasmic reticulums. We have smooth and rough ER. And neurons have this unique structure called a Nissl body, or multiple Nissl bodies, which are rough endoplasmic reticulum that are not directly integrated with the nuclear envelope of the nucleus. So when we were discussing the endoplasmic reticulum before, a majority of the endoplasmic reticulum, its membrane, its internal membrane, is continuous with the nuclear envelope of the nucleus of that cell. So they're basically attached to each other, which allows for really quick transcription and translation through the ribosomes that are on that rough ER. The nucleus, a neuron, the body of a neuron has a lot of free-floating rough ER, which is pretty unique to this cell type. And so under a microscope, that is going to give the body of these neurons a muddier gray color, whereas most axons of our nervous system are going to be surrounded by these films of fat called myelin, which make them look white. And so we are going to be talking a lot about gray matter versus white matter when looking at various structures of the nervous system. Gray matter is where we have high concentrations of the cell bodies, so the receiving ends of these neurons. And then white matter is mainly going to be composed of axons with that fatty myelin surrounding it. So when you hear gray matter, think body, think receiving. And when you hear white matter, think axon, think signal sending. And again, that comes from these Nissl bodies. And so where we're going to be spending most of our time talking about or looking at the neuron is at this axon hillock and the axon itself. So how signals are being sent from one neuron to another. So the axon hillock is referring to this short space between the cell body and then the actual axon itself. And this is going to be an important location for building up what we call graded potentials. So essentially, the overall charge of the membrane at the axon hillock will determine whether or not an action potential, the actual signal of a neuron, is going to travel down the axon. And that's very important because action potentials are all or nothing. If they get triggered, it is going to travel all the way down the axon. As soon as an action potential starts, it is going to finish. There is not much we can do to stop it. And so this axon hillock, determining whether or not that starts, is where most of the control of nervous signaling is going to happen, whether we are exciting or inhibiting those signals. That is mainly going to be happening at the axon hillock. And so directly next to that, we have our initial segment of the axon, which again is where those action potentials are sent. You can see some of these patterns of naming things showing up here. So the axolemma is using the prefix axo to refer to axon, the suffix lemma to refer to the membrane of the axon. So this is the axolemma, which is the membrane of the axon, right? Just like with the sarcolemma, which was the membrane of a muscle cell. And so the neurolemma is going to be the membrane of the cell body and dendrites. Okay, I believe that's everything about our structural components of neurons. And so now taking a closer look at where they meet with a neighboring cell. And again, this could be the next neuron in this pathway, or this could be the target cell on which this neuron is acting. So there's always going to be some space between this axon and its target cell. They are not held onto each other or directly connected like we've seen with things like epithelia. Right? There's no gap junctions, there's no desmosomes holding these synaptic terminals onto that cell. There's always going to be some space. And so that space is referred to as the synaptic cleft. This junction where they meet is called the synapse. So where the axon terminals are going to meet with the target cell, that is the synapse, and there's always going to be a little space between them, that is our synaptic cleft. And so we can refer to the membrane of the axon that is sending the signal as the presynaptic membrane, and then the membrane that is receiving the signal is the postsynaptic membrane. So when we were looking at neuromuscular junctions, the membrane of the axon is the presynaptic membrane, and then the sarcolemma, where there were receptor proteins to attach with that acetylcholine and start a new action potential on the muscle, that is the postsynaptic membrane. And so there are some of our cellular organelles in each of these little synaptic knobs. Got some endoplasmic reticulum, mitochondria for energy. But the most important thing here are these vesicles, right? These little packages full of neurotransmitters. And there may be more than one type in different vesicles in a neuron. Typically, there's mainly one type, if not only one type of chemical being produced here. And then those will be released into the synaptic cleft to cause or be received and cause some signal on the postsynaptic membrane. You can see a little microscopy image of that there. And so as I referred to earlier, that example of a neuron that we were looking at is a multipolar neuron, but that is not the only organization of these structures of a neuron that we have. It is the most common neuron in our central nervous system. It's the most common image that we use when talking about neurons. You've probably seen images that look like that before. But neurons do come in other shapes and sizes. And so the simplest is our anaxonic neuron, which, as the name suggests, they just don't have axons. Right? So this is a cell body with a bunch of dendrites. So it receives a lot of signals, but has no ability to send an action potential. So they can communicate with neighboring cells in other ways through typical like chemical messenger systems, just sending them out into the space to neighboring cells, but they do not use or conduct action potentials. They do not have an axon. And these are pretty uncommon, just found in some of like our special senses like sight, which are, you can find them in the retina of your eyes that help with recognizing like shades and providing depth or three-dimensional awareness. And then the rest of our neuron types are categorized by essentially how many branches are coming off of the body. So we have our bipolar neuron, which has a long branch leading to its dendrites and the typical axon that we were discussing. So we have two poles, bipolar. We have a unipolar neuron, which even though it does branch in both directions, the connection to the cell body itself, there is just one branch. So we have one central connection to the cell body, and then that branch will diverge into two sides, but that is one pole coming off of that cell body. And then we have a multipolar neuron, like I said, the example we were looking at before, where we have all these branches of dendrites coming off of it, and then a long axon. And these are the most common types. So unipolar are typically found in our sensory pathways. Multipolar are typically found in our motor pathways and are the most common. And then these other two are really just found in some special senses. Okay. So neurons are making up our main functional component. When we think of nervous system, you think of signaling, and that's being done by neurons. But that function is being supported by a variety of these ganglia or glial cells. So if you hear the term ganglia or glial cell, that is referring to the supportive cells of the nervous system. And so we're going to have some overlap in function between these supporting glial cells of the central nervous system and the peripheral nervous system. But we're going to have some separation of what types of cells are doing what. So we do have different cell types showing up in these different spaces. Right? In the central nervous system, basically everything that's in that space is the neuron cells, glial cells, and then some blood to help support, or cerebrospinal fluid to help support these things. But basically in the central nervous system, that is all mainly nervous tissue. In the peripheral nervous system, where these neurons have now left the spinal cord or the brain, they are now being interwoven and going between all of these other tissues of your body. They have to fit between your muscles and skeleton and your skin and your organs, right, to go through all these other spaces. And so the organization of the structural network has to look a bit different too, because it's not just being surrounded by other nervous system tissue, it is now being surrounded by all these other spaces that it could be interrupting if it was structured the same. So anyways, we have different glial cells of our central nervous system compared to our peripheral. So in the central nervous system, we have four main types. We have our astrocytes, which make these star-looking shapes. Their main function is to create what is called the blood-brain barrier. So in your central nervous system, a majority of the fluid that is making up essentially the ground substance of our nervous tissue is your cerebrospinal fluid, which is somewhat similar to the plasma of your blood, the fluid of your circulatory system, but it is unique. Right? It does have different concentrations. And the astrocytes are responsible for keeping that cerebrospinal fluid separate from the fluid of our circulatory system. So we do have capillaries and veins that are going to be permeating into the central nervous system, but they are going to have these specialized structures around them created by the astrocytes that could make it much more controlled, like add more selectiveness to the permeability of those membranes. So there's a lot more control about what is entering and leaving the circulatory system when it is in the central nervous system, right? We have this barrier that controls the diffusion and absorption of nutrients in and out of the circulatory system to the cerebrospinal fluid, etc., and back and forth. So that's mainly what astrocytes do. They also clean up the excess neurotransmitters once they have, already sent their signal at the end of that synaptic terminal. So they'll help pick those back up and get them back into the synaptic knobs of the previous neuron. Oligodendrocytes, big scary name, are here to facilitate the speed of signal conduction. So these are these little bubbly, knobby looking cells. These are oligodendrocytes. Their job is to myelinate axons. So myelination is referring to this fatty sheath, essentially, that can be wrapped around axons. And that's going to be really helpful with speeding up action potentials. We're going to talk about the specifics of that here in a little bit. But essentially what this is doing is locking in whatever ions and molecules have entered the cell at the axon terminal or at a internode, at a node itself. So whatever ions have flooded into this node, they are now basically locked into the cell, cannot exit the cell until they get to the next node. So if we had an action potential starting here, all of those ions that have now flooded into the cell are going to keep dispersing throughout the axon until they reach this next node. And now we can have another essentially action potential continuation. The more voltage-gated ion channels are opening, more ions are flooding in, et cetera, et cetera. This just speeds up the process. So myelination is adding a fatty sheath to the axons of a neuron in order to speed up the rate of action potentials. And this is what makes white matter look white. So again, white matter is mainly myelinated axons like this. And that's from oligodendrocytes. Our third glial cell of the central nervous system are microglia. And these are just the macrophages of the central nervous system, your immune system cells. Right? They're there to protect, destroy. And lastly, we have ependymal cells, which are basically going to be lining any of the open cavities in your central nervous system. And they are responsible for producing all of our cerebrospinal fluid. There are a few specific areas where these ependymal cells have been specialized to do just that. Those are called our choroid plexi, or a singular choroid plexus. So those choroid plexi are what produce cerebrospinal fluid, which is the fluid of the nervous system. And then all of the open spaces in your brain are called ventricles, and then in your spinal cord are called the central canal, and ependymal cells are lining those spaces. So those are the glial cells of the central nervous system. And here you can see those ependymal cells lining this central canal. So this is our first look at the actual spinal cord itself. So if you think about the structure of a vertebrae, the bone that would be surrounding your spinal cord, remember that vertebral foramen, the space in the center? This is how your neurons are organized in that space. Right? So this is a transverse section of the spinal cord. If you were to cut through like that, remember transverse is this cut. This is what you would see. And so in the very center, you have this hole, which is your central canal, and that's full of cerebrospinal fluid. Here's those ependymal cells that completely line that space. And you can see that they're surrounded by what we call gray matter, which are going to be the bodies of neurons. And then those neurons will have axons that are going to move out into the white matter and make that look white because of all of the myelinated axons in that space. Okay. And so we have different glial cells of the peripheral nervous system. And mainly, we have these, or what we are concerned with are the Schwann cells. There are also satellite cells, which basically do everything else, or they're kind of the astrocytes of the peripheral nervous system. Satellite cells are also what get essentially absorbed by muscle when it has experienced hypertrophy and now is going to recover and grow. They absorb satellite cells, and those become part of that multinucleated skeletal muscle cell. But we're mainly concerned with Schwann cells, which are the oligodendrocytes of the peripheral nervous system. This is what is myelinating the axons of the neurons that have left your central nervous system, right? They're now in the periphery. And Schwann cells do that a little bit differently than our oligodendrocytes. So if I back up a bit, you can see that one oligodendrocyte here is myelinating two different axons, so the axons of two neurons. This is another oligodendrocyte, and you can see it's myelinating two different parts of the same axon. So in the central nervous system, one oligodendrocyte can myelinate multiple axons. In the peripheral nervous system, each Schwann cell that is myelinating an axon is only myelinating one node, one internode, of one neuron. So every single segment you see here where we have myelination of this neuron, that is a separate Schwann cell. Okay. So these Schwann cells are much more specific in their function, only myelinating one neuron, and they completely wrap their entire cytoplasm, the entire membrane around that internode. Whereas the oligodendrocyte would have its cell body somewhere in the fluid, the space around it, and just have extensions of its membrane and cytoplasm covering these Schwann cells completely surround it with their body basically attached. So when you have it organized this way with one Schwann cell on one segment of one neuron, that is a myelinated axon of a periphery. But then we also have Schwann cells that can have multiple axons around it, and these are typically for much thinner axons in diameter, so smaller neurons with a much thinner diameter axon, and these are considered unmyelinated axons. It's not really providing that same function of saltatory propagation that we'll talk about with myelination. So these are not fully myelinated. These are considered unmyelinated axons. So when it is 1 to 1 to 1, you get a myelinated axon. If there's one Schwann cell with multiple neurons or multiple axons surrounding it, that is considered unmyelinated. Okay, and so I believe this is where I want to stop this video. Yes. Okay. So that was your introduction to the different divisions of the nervous system, your introduction to the cell types of the nervous system, and then in the second part of this video, we'll be talking about essentially nervous signaling, action potentials, and how all that stuff works. So I'll see you in the next one.